Hi everyone, welcome to the workshop. It's the fourth time that we're having a workshop which is completely dedicated to theory in neuroscience. And our first speaker will be Idan Segev.
Thank you, Yaron. Thank you all for coming. I think this is a very good idea to have kind of a long talk, a long discussion, enables us to speak our minds about what I'm going to say. I'll be very happy to get feedback because, as you will see, some of the things that we understood at the biophysical level. Some of the things that we should understand at the more computational levels are still waiting for good ideas, hopefully from you. And also, I think the whole notion of having a focused theoretical work or session, because many, even at ELSC, still are not very much aware of the fact that we need very much today, with all this bombardment of data, we need some theoretical framework, and especially combination of experimentalists and theoreticians working together, very, very essential, I think, for the development of the field. So I hope this will be a place for linking theory and experiments, as I will try to show you today, that I was very much inspired myself and our students, my students, in this case, Albert Gidon and Lital Bar-Ilan, whose major work are presented here. Some of it was recently published in Neuron in Frontiers in Computational Neuroscience. Some of it are in progress. So I will try to summarize what we did in a perspective that is somewhat different than the perspective that is in these papers. So I want to really start, as I like to start my talk, with some history about inhibition. I'm going to talk to you about inhibition today. So I is inhibition. It's not I, it's I, inhibition. We are talking about inhibition today. Then I will raise these 6 questions that came along my mind while I was looking at inhibition deeply in the cortex mostly. So these 6 Ys on the dendritic eye came from looking at inhibition more and more, learning about inhibition in the cortex, and then these 6 questions came about, and then there was this development of, as I see it, as a new, one can call it theory, of Albert mostly, about how to analyze dendritic inhibition, and then I will provide some surprises that came through this work. And finally, if I have time, I'm not sure if I have time to get into the number 6, I want to speak about inhibition on dendritic spines, which is a relatively new subject for me. So you know the father of inhibition is this guy, this fellow, Sir Charles Sherrington. He devoted his whole Nobel lecture about 80 years ago to discuss inhibition. So the whole whoever is interested in inhibition, he certainly should read the Nobel lecture by Sherrington. And already there he said several things that some of them really still influence us a lot today. Of course, there are many, many new concepts, especially coming from theory. But he already then spoke, you know, mostly Sherrington was dealing with spinal cord and the balance between excitation and the coordination between excitation and inhibition. And he speaks about about the interaction between the two, and he spoke, of course, and he said eventually the excitation inhibition, the one is no less important than the other. Here he speaks about the balance between the two, and then he further said that the whole operation of the nervous system. It's amazing that 8 years ago it was not at all clear that there are these two major systems in the brain, inhibition versus excitation. He was basically the one who really put it on the table at the level of synapses. So he spoke about the co-adjustment of excitation, inhibition upon individual units. He speaks about muscle units, so the muscle unit should coordinate itself in order to function by having a balance or co-adjustment of excitation, inhibition. Certainly not the balance that, for example, Chaim and Carl and David are talking about. This is a different type of balance, but still the notion that if one goes out of balance, the system goes into a state that you don't want, and if the other one goes out of balance, there is another state, so it has to be co-adjustment. He said there's something interesting, when we talk a little bit about that later, that even today we think about inhibition, at least some of us think about inhibition, always how it affects excitation. So there is no inhibition on its own, because you think that the excitation is the major thing, so to speak, the output, the excitation, and the inhibition is always with reference to excitation. So you define inhibition with reference to excitation in some sense. At least that's what he said in his third point. This is a very, very, very crude summary, of course, of many, many, many pages of his lecture. And finally, he speaks about that excitation propagates, yes, and inhibition does not travel. So there is a difference between the two. Excitation goes from one cell to the other. If it succeeds to activate the next cell, inhibition does not travel. That's how we thought about this. So that's old way of thinking about this, but at least this is the history of thinking about inhibition and excitation 80 years ago. And then the first time that anybody introduced the notion of dendritic inhibition, the term dendritic inhibition came in this paper from Rodolfo Inas, working on the cerebellum. And he was the first basically to reconstruct presynaptic cells and the postsynaptic Purkinje cell. And he was the one, as you see here, to show that some type of inhibitory cells tends to particularly, specifically target sub-region of the dendritic tree. And you will see later that this has became a common knowledge, that inhibition is sharing the labor, so to speak, and some region of the dendritic tree is occupied by a particular subset of inhibitory neurons, and another subset by another type of inhibitory neurons, and that's, of course, one of the puzzles. So this is the first time that we hear the notion dendritic inhibition. So I want to go about the six whys about dendritic eye, the sixth question about dendritic inhibition, but before that, just another introduction. And this comes from the Human Brain Project, the work that we are doing with Henry Markram, that I'm not going to go into it now, the whole project, but just to tell you what I learned about inhibition from this project, because there is a very, very systematic attempt in this project to classify and to put in order the data that we have from diverse labs and diverse experiments into some kind of a framework. So, of course, this is, again, very brief, just to let you know that working with Henry and with Javier de Felipe, who is an anatomist, and other people trying to make sense of the cortical column, so to speak, trying to characterize the type of cells there, defining the column, and all these things that are hard to define, and I'm not going to go into it, just to tell you that one of the aspects of this project I'm not involved in, this particular subject project, but it's almost done, is the attempt to classify, in terms of the anatomy of neurons, the type of neurons, anatomical types in the cortex. So what you see here is the different 56 morphological types that were characterized. It's not published yet, but characterized systematically, using classification methods and other methods that were characterized in the cortex, neocortex of the mouse in this case, of the rat in this case. And so if you look, for example, at layer 1, you see that layer 1 consists of only inhibitory neurons. There are no excitatory neurons in layer 1. And you can see in layer 2, you can see in layer 4, you can see in layer 5, and so forth, all these types. And already I highlight now, a question that will come later in another perspective, is why do you have so many morphological types of inhibition in the cortex, so many, in terms of types, not in terms of numbers, in terms of types. Why do you have so many inhibitory types and so few, so to speak, excitatory types? It's not a priori clear. Why do you put, and you will see later, that this population is about 10%, between 10 to 12% in the cortex are inhibitory cells, GABAergic cells, and about 85 or 90% are excitatory glutamatergic cells in the cortex. But the majority, in terms of number of types, although few in percentage, a lot of types. This is morphological types.
Can you explain what the pictures represent?
Yeah, okay, so the picture, so this is like a cloud. Each cell here is a cloud, as you can see, And so this is one example of a cloud. So you take, let's say, for example, layer 5 pyramid, 6 tufted pyramidal cell. This is, I don't know, this cell, I think. Yes, in layer 5 there is 6 tufted pyramidal, this one. And you take all the cells that you have, about 100 or several hundred that were reconstructed, you put them all on this axis, on this XY coordinate, 0 coordinate, which is the cell body, and this generates a cloud of statistical neurons, statistical morphologies. So this is, if you put all the 200 layer 5 pyramidal cells, you will see that the dendritic tree occupies this cloud. Yes? And the axonal tree, so this is the dendritic cloud of many, many cells of this type. This is the dendritic cloud of many, many cells of this type. And this is the axonal cloud to the left. To the left is the axonal cloud when you put them all together. So we try to just characterize the space within this column, and I didn't get to define what this column exactly, just to see how the distribution and variability and the variance and all that morphologically, it's only morphological, yes? It's like you go in the jungle and you put all the trees of the specific type one on top of the other, and of course they are not exactly the same, so you get the cloud. So these are the clouds. So I don't want to go into the details, just to highlight the fact that there are so many morphological inhibitory types in the cortex. Maybe later I will also mention, and I'm sure you know, also a lot of chemically distinct differences, for example, somatostatin and parabulmin and other chemicals that are being generated, so you can classify these neurons also by the chemicals that they produce. You can classify these neurons by the genes that are expressed and so forth. But it's interesting to highlight, and that's a point to discuss later, why do we have so many, in this case, morphological types? You will see also chemical types and so forth, particularly in the inhibitory class, and less so in the excitatory class. Why?
Is there some regularity in the way these are distributed in the cortex?
So yes, but let's wait a little bit, but because here I don't tell you anything about the percentage of each one of them, yes? It could be that this is the major one, the basket cell, and these are very few and so forth. All this is part of the human brain project. The whole notion of this part of the human brain project is to make sense, to classify, to put in order. In Bal asked me before, after speaking with Henny, how do we go about putting data in order, medical data, and in this case, neuron data. How do we put it in order? So this is one example. By the way, paper in Nature, huge paper in Nature is about to be coming in a few months, summarizing all that, the whole concept of putting data together. This is morphological data. You will see soon. You will see electrical data with genetic data, chemical data, to put it in order. So I'm just highlighting what we know today.
Yeah. How distinct are these types?
Sorry?
How distinct are these types?
So this is all. This classification are done objectively. You throw all these cell types into a pool, into a basket, and you use modern classification techniques to objectively say that this cell is sufficiently different than this cell morphologically to say that these are two types, or this is enough different from this cell. So layers comes automatically and so on. So again, this is not the subject of my talk now. I just wanted to highlight the different types of cells that you cannot see here. Of course, there are a lot of new techniques, as you may know, to stain the different chemicals, so to speak, and so you can see the different cell types, in this case inhibitory versus excitatory. You can stain for GABA, you can stain for all cells, then you can subtract one from the other, and then you can see all the inhibitory neurons separately as a group, as a whole group. And then you can do things like that, for example, which is part of this paper that I'm telling you about that will be coming. It only gives you percentage of inhibition per layer, for example. In this case, you will see that 100% per layer 1 are inhibitory, 20% per layer 2 are inhibitory, and so forth. And then you can also divide it to subclasses, because I can tell you today of this type that I showed you, what is the percentage of this particular anatomical type in this layer 3. So I can tell you, the basket cell is 5% of all inhibitory neurons of this type and so on. But this is not what I want to tell you. I just want to tell you that we are getting a major catalog of cell types within cortex, and this catalog consists of morphological types, anatomical types, genetic types, per layer, which will be the basis for later simulating the cortex which I'm not going to talk about now. I'm just telling you of the interesting thing is that the inhibitory types, as you will see also later electrically, are very, very rich, although there are few in percentage-wise. Okay, so this was an introduction about inhibition in the cortex in this case. Now,
short question. The layers are really discrete, or do you see continuous transition between the layers?
well, I mean, you know that layers were defined already by Harmonica Hall.
Right.
I must tell you that, for example, when I look at it, I'm very bad with looking at layers. I don't see that there is a distinction between here and here, but I must say, by cell body, this is a stain for cell body. But this was the distinction, that this is layer 1 and there is less cells here, and then there is another layer 2 here. But as I said, doing this objective classification, the layers comes up without you telling them that this is layer 2, 3. Yes? So there must be something about the layers. But again, I'm not an anatomist. I'm just telling you that looking at this, I'm learning something about the richness, the diversity, the interest of this philharmonic music of anatomical, electrical, and all these other musical things. And so eventually we will come with some kind of a, I think, compact description as possible of who are the characters, the anatomical characters, the genetic characters that build up a column. What should we do with this? This is the major question of this talk, focusing on inhibition. We also have today another important information that comes from this very, very, very elaborate Blue Brain project data management, is also to speak a little bit about the distribution of different types of inhibitory cells. For example, on layer 2-3 cell, this is layer 2-3 pyramidal cell, that how do the different anatomical types that I mentioned before in this layer, particular layer, converge into the dendrites. So let's say the red circles are synapses coming from Martinotti cells. The blue circles, let's say, are synapses impinging on this dendritic tree from basket cells. So you have also this kind of a synaptic map, inhibitory synapses. These are all inhibitory synapses. So this information is a combination from reconstruction of pair of neurons, inhibitory and layer 2-3, inhibitory and layer 2-3, inhibitory and layer 2-3, pair reconstructions, many, many pairs. I think there are about 70 pairs of layer 2-3 and the presynaptic inhibitory cell. And also, a lot of it is coming just after putting the column together, as Henry likes to put things together, physically putting together in the computer, and then you have some predictions about where the axon of this cell that was never really actually fully reconstructed should touch putatively here or there. So some of this information comes from actually pair recording and anatomical reconstruction from inhibition to this type of layer 2-3 cells, and some of these locations come from just predictions about where the axon of this one cell should impinge with closer position to the dendritic tree. But again, you can see that we are getting slowly a map of synapses, in this case inhibitory synapses, on the postsynaptic target.
The case was constructed with one pair, so this is inhibition coming from the ELSC.
No, this some of these are coming, no. So I made a mistake. I think I made a mistake. I said that the presynaptic cells are all from layer 2-3. No. This is layer 2-3, and this is coming from all layers in the column. So, inhibitory cells are mostly local. Some of the axons are more global. So most of the synapses here, inhibitory synapses here, are coming from layer 2-3. Some of them are coming from other layers. For example, layer 1 synapses project, inhibitory synapses project to layer 2-3. So some of these synapses might come from these 6 cells, inhibitory cells coming from there. So this is the total predicted, so to speak, type of cells impinging on these inhibitory cells, impinging on this layer 2-3, not only from layer 2-3.
Which indicates that Sherrington's wrong, that inhibition does travel.
That Sherrington is wrong?
Inhibition does travel.
Well, he meant about electrical traveling, not traveling anatomically, but electrical traveling, that I inhibit you and you inhibit him. And that's what he said cannot happen, because in order for you to inhibit him, you should be excited, not inhibit. That's what he meant about propagation. So not anatomical propagation, so to speak, but electrical.
You left out the chandelier cells, or what?
The what? No. Oh, you mean chandelier. That's a very good point. Very good. Okay, chandelier. So chandelier cells, this cell, okay? This is a reconstruction. This is a beautiful work. So as I said, much of our work comes from Javier de Felipe, who is a really great anatomist and very careful anatomist. And for example, you can see here the chandelier cells that are so particularly unique that send their axon only and specifically to the initial segment of the axon here. Only. This is the most specific cell in the brain that we know in terms of connectivity, I mean. Yes, the most specific cell. And this does not come when you put the axons together in the Blue Brain project. So statistically, if you put all the cells correctly and the chandelier here and the axon there, they don't go only here. You have to have some kind of a learning rule that direct the axon only there. We have a lot of other problems. Again, I don't want to go into it because some, of course, of the axon go through the soma if you just put them in space. And you have to avoid it. So you have to have some algorithm to avoid soma or avoid dendrite sometimes, maybe if you want to go through, otherwise you're stuck. So there are all these issues about how putting things together, but I don't want to talk about this. What I wanted to talk about the 6 Ys on the dendritic eye, eventually. So this is the first I, Y. This is the first Y. Oh, there was another Y that I forgot. This was the first one. The first one was, the most basic one is, why does inhibition cover the whole dendritic tree? In principle, because again, talking about chandelier, if you want to veto the output, if you want to control the output, particularly the output, you should put all your inhibition near the cell body, near the axon, naively thinking. Why should I put inhibition here, inhibition there? It's a waste of inhibition, because eventually, if your role in life is to control the spike output, and the sodium spike output in the axon, just veto it in the axon. So this was my question.
So why excitation? Why do we need to do three?
That's a question. Yes, I'm talking about inhibition. We can ask the 6, actually more than 6 about excitation. But no, particularly because we think about excitation. Unlike excitation, we think about inhibition naively. I want to think differently, but I'm saying naively we thought about inhibition as trying to really control only the local output.
Why not excitation?
Because excitation is for another talk, but excitation, I can think about excitation as trying to bring physically a lot of inputs from many sources.
Control the sodium out.
Yes, but if you want to have, again, I'm not talking...
Also inhibition brings information.
No, but the role of inhibition is to stop excitation. The role of excitation is to bring information.
Generate sodium spike, which is a soma.
No, but again, I don't know if this is the reason, but I can think about thinking like this. Suppose I want to bring in inputs from many excitatory sources. I cannot put all these I don't have space to put all these excitatory sources, let's say in the axon or in the soma, because I want 10,000 synapses to impinge on and so this will bring me excitatory information from there, and this will bring me excitatory information from there. Let's say that we think like this. So I need the whole dendritic tree to collect excitatory information. But in terms of inhibition, it says one thing.
You're saying if information is excitation and inhibition is just a break.
Yes, that's what I'm saying. That if I'm thinking like
inhibition and information.
Yeah, I'm saying that if I'm thinking like Sherrington, and I'm thinking.
One is not more important than the other, right? So
okay. If you insist on asking the same why excitation targets the whole dendritic surface, it's a good question, but for another talk. The other question is, why do we need all these subclasses? So many subclasses. I showed you only anatomical subclasses, but you will see also electrical subclasses. Why do we need all these subclasses covering or converging on particular sub-tree? So this is a summary of Kevin Martin and Robert Douglas. So you just saw the chandelier targeting only here, particularly. As I said, the most specific target cell that I know of is this chandelier, but you know that the basket cell tend to target the basal tree mostly in this region. The double bouquet you can see, You don't see the Martinotti tends to go there. So there are all these subtypes sharing the labor in some sense. Each one is occupying, each one is targeting different subtrees. So that's my second question. Why is this subdivision of labor? This is also true for the hippocampus. This subdivision of labor of different inhibitory cell types. We don't know about excitation, actually. I don't know if this subdivision is, as much as I know today, nobody showed this, that there is a particular subdivision in the hippocampus. Yes, excitation of different layers do come to different regions in the cortex. I don't know exactly if there is this subdivision of excitation, but inhibition is very, very clear. Very, very clear. That at least from all these works, from Somogy and others, you can see, the chandelier going here, and you can see other cell types going there, but particularly there. Not statistically there, particularly there. Very, 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 very clear. Another interesting question number 3 is why a single inhibitory axon makes so many contacts on the postsynaptic target cell. So for example, the excitatory neurons, at least this I know, excitatory neurons, let's say layer 5 to layer 5 excitatory neurons, pyramidal cells, are making on average 5 contacts per axon. So if I'm an axon, I make 5 contacts on you, if you are the same type of process, let's say layer 5 pyramidal cell. Inhibitory cells tend to make much more than excitatory cells contacts, inhibitory contacts, per axon on the postsynaptic dendritic tree. Much more. Between 12 to 20. Why? Why, if I'm an inhibitory neuron, I want to make absolutely sure that if I contact you, I may not contact you, but if I contact you, I make 12 or 20 synaptic contacts with you.
This is on the same dendritic branch, or?
On the same territory, on my domain. So if I'm a chandelier cell, I'm going to your axon, and I'm making 12 contacts there. If I'm basket cell, I'm going to your basal dendrite, and I'm making 15 contacts there. So on my domain. But many. Oops.
No, but if you have 2 domains, it's times 2, or is it split between the 2 domains?
Total per axon. So if I count the number of contacts that I make while passing through your tree, is between 12 to 20.
So again, how many total inhibitory synapses?
In terms of percentage?
Numbers.
Number of synapses per what? Per cell?
Per cell.
About 10%. Both the number of synapses per cell are 10%, inhibitory synapses per cell. On average, it's 10%, but also the number of inhibitory cells per column is 10%. That means that many cells do not get inhibition. But when you get inhibition, you get a lot. Question number 4, and this is an electrical question. And as you will see, I don't have good idea about this question, about the answer to this question. If you characterize, this is a paper that came with Charles Druckmann. I forgot to mention him. The work with Charles Druckmann was a work that was published recently in PLOS Computational Biology about characterization of the different electrical types, not morphological types, electrical types of inhibitory neurons. Okay, so you put, again, in this case, not the morphologies, but the firing properties of the different cell types. These firing properties are characterized following a step current pulse. So you give this cell, let's say this, I don't know what, basket cell, you give a square pulse, current, and you get this firing property. Another cell type, electrical type, will fire like this, and like this, and like this, and like this. And the question is, and this is a very classical old question, Mike Gutnick, for example, was involved in that, in the nomenclature, trying to find a nomenclature for the different types of firing properties or firing repertoire of cells in cortex in general. So you know the classical, adapting cell, bursting cell, stutterer cell, regular firing cells. All these terms came from intuitions of many people sitting and looking at it. What we did with Shaul is to take all the data that we have. This is a huge amount of recordings. I mean, this is really, really, really many millions of spikes, putting them in baskets and asking, are they really different? are there really subclasses, electrical subclasses? And I can tell you now astutely that if you compare the firing properties using this exactly the same method for excitatory cells and for inhibitory cells, so this is now only the inhibitory cells, there is a huge difference in terms of the richness of repertoire of firing. The inhibitory cells are much, much more rich in terms of the variability or variance of the repertoire of firing of inhibitory cells compared to excitatory cells. Excitatory cells are relatively stupid, dull in their firing properties. Inhibitory cells tend to have a very much rich music of firing. Why? Why do you put in such a small number, percentage-wise, of cells, the richness of firing capabilities, meaning different ion channel types in the memory? Why you insist why the designer insists to embed on the different cell types, anatomical cell types that I mentioned before, also this richness? So you may speak, and this is part of the Nature paper, you may speak of morpho-electrical types. Because each morphological type, let's say basket cell, may have several electrical types. So how many total morpho-electrical types do you have? It's about 250, but again, I don't want to go into it in terms of an inhibitory cell. I just want to highlight a question. I'm saying that the inhibitory cells are rich in their firing repertoire. Objectively, they are rich, meaning that the channels that they express richness of firing more than the excitatory cells.
I don't know if it's the property of the network or the school of...
No, sorry, this is individual cells recorded individually out of the network. Each cell here, each line here is a cell in the dish, without synaptic inputs, isolated. You record, you inject square pulse, also sine wave and all that. So all these characterizations are for individual cells, not in the network. You could ask a good question, whether these characteristics remain in the network. This is a good question, but I'm not dealing with this question now.
How confident are you that these firing types, these intrinsic firing types, are don't shift in a given neuron?
I'm not confident. I'm saying I'm not confident at all. I know that this is affected by hormones, as you know very well. It's affected by other environmental issues. I'm saying that if you take systematically again and again, the same cell type, and you do this characterization, as I said, I have millions of spikes repeatedly for many hundreds of cells, you get this richness, which means that something embedded in me, the richness is embedded in me. I'm not saying that I'm always myself, under other circumstances I'm not Idan anymore. But at least in this case, because it is repeated so many times and because of this statistic, I'm now confident that
But if you take a given morphological type, you can get more than one of these.
Yes, I just said it.
So it's possible that...
I just said that basket cells,
like this,
I just said that this is mapped onto the anatomy, not one-to-one. I just said that basket cells can express it. So I'm saying that basket cell is an anatomical name, and bursting cell is an electrical name, and the question is how many combined names do you have? I said about 250, but again, I don't want to go into it because I won't go into the lecture. This is not the lecture. This is not the question. I want to give the answers too. But anyway, so you understand the type of questions. I'm looking at this population. It's a very interesting population. Poor inhibitory neurons, poor in percentage, rich in other things. So I'm trying to understand some of it. Also, this I would like to understand. Why do you need so little percentage-wise? Why not 50-50? Now, the question, of course, how do you count this? We may discuss with David. Is it intrinsic into the cortex? Because most of the excitatory cells, axons are coming from the outside, and these are mostly local. The inhibitory cells, synapses are mostly local. The excitatory synapses per neuron are mostly from the outside. So, but in terms of if you count per neuron or per colon, if you just count the number of synapses, symmetric versus non-symmetric, inhibitory versus excitatory, you get this number. This number, by the way, is getting down with time as people are doing more and more systematic. It's about 10 to 12% today in the cortex of the mouse P14. I'm don't know what happens to human, but now Javier De Felipe is looking at human cells, reconstructed human cells, and it's about the same percentage. So less inhibition, total, both in terms of number of cells per volume, and also in terms of number of synapses per cell.
Does this mean that a given inhibitory cell has less targets than an excitatory
Yes.
cell?
Mm-hmm. Otherwise, you cannot keep both the number of
contacts.
Yeah, exactly. That's what I said before. You either make sure that you contact the postsynaptic cell, then you make many synapses, or you don't care and you don't contact the other cell. Otherwise, it's not consistent. Okay, what was my, this is my 5, and this is my 6th and last question. And this is a recent thing that I'm thinking about with Albert, is the following. We knew for many years, many years actually, since the first EM reconstruction of cortical neurons, that sometimes on that on a dendritic spine, so this is a dendritic spine, that in almost every dendritic spine will have an excitatory synapse onto the spine. So this is the excitatory synapse onto this spine, there is another excitatory synapse into the other side. But some spines, some dendritic spines, that, as I said, always or almost always get excitatory synapse, also sometimes get inhibitory synapse. But from recent results, we know that in terms of percentage of inhibitory synapses that converge onto dendritic spines, it's much higher than the just by chance, covering the tree. That means that not only the excitatory synapses tend to target particularly, specifically, the spine, because you don't have many excitatory synapses on the base. Most of the excitatory synapses on pyramidal frames are on spine, but also a high percentage of the low percentage that you have of inhibition, about 30% of inhibition goes to spine. Then it's together with excitation.
How many spines? What's the fraction of spines?
Number of spines is like number of excitatory synapses.
Yeah, what is the fraction of spines that also have inhibition?
30, the fraction of spine, very small. Very small percent. Something on the order of 7 or 6%. The fraction of spine that have this arrangement. But it's, in terms of percentage per inhibition, it's much more than expected, just based on distribution. So there is some kind of a targeting that we are trying to understand, yes? Of course, you may ask, as Chaim asked me before, but you may also need to explain why excitatory synapses are going to the spine and not here. Which is also a very classical question. Why do excitatory synapses do not make contacts here, mostly? Of course, most of the inhibitory synapses are on the dendritic shaft. Most of the inhibitory synapses are here, so about 70% are here, but some of them are also here. So that's the last question, which I don't know if
Are there also dendrites a spine with only inhibitory synapses?
Essentially, no. Essentially, no. There is one thing, maybe I'm jumping, but there is one clear thing, because as I said before, a single inhibitory axon is making many synapses per this neuron, as I said before. So it has this property. It goes, make contact on spine, let's say, then it's always together with the excitatory synapse, so this is inhibitory excitatory, together on the spine. Then the same axon is making synapses on the shaft, elsewhere on the same neuron. So it's not that this axon makes sure that it always makes only on the spine. This is not the case. The case is that when it makes on the spine, it typically makes not on the spine, in the same axon, in the same postsynaptic neuron. Anyway, these are the questions. I will go very fast. So you know that we are, in order to understand current flow, voltage flow, in this case I will show you conductance flow in dendrites, we are using the cable equations, and we are taking the elaborate dendritic tree, reconstructed dendritic tree, breaking it into cylinders, into cables, and we can compute analytically, as you may know, we can compute analytically the spread of voltage assuming that the dendrites are passive. You can use the passive linear cable equation to compute the spread of voltage or current in the tree. And so I'm going to jump on this because this is classical books just for the students. I want to tell you that in this case I will show you that we solved the cable equations for a new measure that is the conductance change, the spread of conductance change, not of voltage or current. the spread of conductance change in dendritic cables using the cable equation and what we know about the boundary conditions. So because I don't have time, I'm jumping on the mathematics a little bit because we understand very well. We developed intuition due to Wilfrid Roll cable equation and voltage distribution about how the boundary properties or boundary conditions in the dendritic tree affect the spread of voltage, whether it's an infinite cylinder or whether it's a short sealed end cylinder. We understand this, but what we try to do with Albert is to solve the cable equations for a new measure, a new parameter that we call the shunt level. So this is voltage, and you know that voltage is very much affected like any other spread on, as I said, the boundary conditions. So if the current flow this direction, you have to solve this piece of cable assuming a leaky end boundary condition because current can flow here, and this leaky end boundary condition depends on the morphology and electrical properties at the branch point and so on. So these are classical work that I thought I would have kind of an introduction to students, or some of you who may did not look at the cable equation, but they don't have much time, but maybe later I will come back to voltage, just to tell you what we know about conductance. So the classical work about conductance was done basically about dendritic conductance. The spread of dendritic conductance in cylindrical cable was done by Christoph Koch and Tommy Polcio in the 80s. And the general notion is that a conductance change, when you have a local conductance change, because you know synapses do not inject current, synapses perturb the postsynaptic cell by opening conductances, and so this postsynaptic cell or cable now is perturbed because there is a conductance change here, there is a shunt here due to the synapse, and the question is how this shunt spreads. How do you see this shunt? Shunt will not really spread, but when you measure it in one location, let's say here, you may detect the conductance change here may be visible here. But the idea was that this visibility is very short, so that the conductance spread, the visibility of conductance is very, very narrow, very, very local. So if you measure here, or if you measure there, you won't see anymore the effect of the conductance change, not of the voltage change, the conductance change. And indeed, some measurements for a single perturbation, so you can dynamically clamp the axon here. You can use an electrode locally and imitate and mimic a conductance change, and you can measure the spread of the visibility of conductance change with distance from the perturbation point and visibility of voltage change from the voltage perturbation. You can see that the conductance tends to disappear very fast in terms of visibility. So if you go a little bit away from the synapse, you won't detect the conductance change. You inject current here, this current should go there, should detect the conductance change, should go here in terms of you measure voltage. So this is why, and I can mathematically show you.
But why is there changing the conductance? Is it because the membrane is active, or?
No, because the synapse is active. So the synapse open, let's say GABAergic synapse. Let's say silent synapse without a voltage, just changing the G synapse is increased. Because of the increase in G synapse, you will have a local shunt. And so there will be a change. What?
It's a change in conductance.
It's a change in local conductance, yes.
Okay.
But this change can be detected if you measure, if you inject current here, and you want to measure the change here, you may detect. So you measure, let's say you measure input resistance. You inject current here, you get an input resistance of 100 megaohm, and then you do a little shunt here, and this 100 megaohm will drop.
Okay.
This is what I call visibility.
Not because the conductance of the membrane between the point that they're changing.
No. Just because you inject current here, and this current goes there, and it's being shunted a little bit by the new conductance change, you will feel here a drop in voltage.
So you measure the global conductance?
The visibility of conductance for the global. It's a local conductance change. It's not that the conductance spread, but the conductance affects other sources that are not necessarily near it. But what I'm saying is that because of this attenuation, I'll show you mathematically in a second if I will get to there. Okay, if I will get to there, just to tell you, so there are these two common knowledge. The local conductance change is very local, and that the synapse, inhibitory synapse, is most effective from the soma point of view on what Koch called on-path condition. So inhibitory synapse, when it sits between the excitatory synapse and the cell body, is much more effective on shunting the excitatory current when the excitatory synapse is more distal from the inhibitory synapse compared to the same excitatory synapse sitting here. So this excitatory synapse, and sitting at the soma, this excitatory synapse will lose a little bit of current backward to the shunt, but most of the EPST will be seen here for this synapse. And if this synapse is active together with this synapse will veto or partially veto the current flow, excitatory current flow to the soma. That's another, what is called the on-path condition. Inhibition should be on the path between excitation to the soma. So this is two common knowledge. And this was proven mathematically by Koch and Poggio. So I want to introduce very fast this term, the shunt level, which is after I explained just before what I meant about to run, what I meant about visibility of the shunt. So I want to define the shunt level, which is a percentage drop in input resistance at some location D, following a perturbation, a conductance perturbation at some location I. So I perturb the membrane here, shunt the membrane here. There is no voltage, so it's just silent inhibition, yes? Just shunting locally the membrane, and I measure the change, percentage change of input resistance here, following the conductance change there. So I gave you an example. Let's say that I measure input resistance here, which is 100 mega ohm before activating the synapse. Now I activate this shunt, steady state shunt. The drop of, now I measure input resistance here, which is now 80 mega ohm. So I would say that the shunt level is the change in input resistance, which is 20 mega ohm, divided by the original input resistance, which is 100 mega ohm. So the shunt level will be 0.2. Okay, that's the definition of the shunt level. It's a natural definition because you want to understand what is the effect of a synapse on the voltage drop at a given location due to the activation of the synapse there, and you can see immediately that the change in voltage, steady state voltage, following an injection of current here, relative to the voltage before the perturbation is exactly the shunt level. So if I say the shunt level is 0.2, I'm telling you that at this location, the steady state voltage will drop by 20% following the shunt. And because of this, I can solve the cable equation, because I know to solve the cable equation for voltage, and so I also know to solve the cable equation for proportion, for a change, for a drop in voltage. And that's the whole idea of solving the shunt level for a cable using the cable equation.
The previous slide had a graph showing quite a difference between voltage and conductance.
Yeah.
Can you relate this?
Yes. So you can mathematically show, again, I don't want to go into it, but later we can discuss it. You can mathematically show, because of this proportion, that the shunt level, the drop of shunt, the spread of shunt level, the spread of the shunt from its location to another recorded location, the drop of the shunt, goes like the multiplication of the voltage drop from here to here, multiplied by the voltage drop from here to here. That's what is written here. And that's why the shunt, the spread of shunt is always more steep, because it's a multiplication of two things that are both smaller than one. So, and this I can prove mathematically, but I don't want to go into it, but we understand why the shunt is less visible than the voltage. So if this was a voltage change, I will see the voltage change here percentage-wise better than if I will shunt this region and I will record the thing here. Also, there is some other interesting things that I don't want to go into it. You can see now that the shunt level is symmetrical. It doesn't matter if I shunt here or record there, or if I record here and shunt there. Because of this, the shunt level, it's like the transfer resistance. That Rij is equal to Rji. That's exactly the same reason. But the voltage spread from here to here is not symmetrical to the voltage spread from here to here. So anyway, so there are these other properties. I don't want to go into it. So let me go into some results now fast because we can solve for any passive dendritic tree. We can solve analytically, assuming that there is a steady state conductance change here. I can look at the spread of the conductance, the spread of this shunt level from its input side to other trees. The dotted line is the classical roll computation for this case. The dashed line, you can see that the voltage drop, if I would inject current, linear current, steady state current here, I will get this huge voltage drop here and a very shallow voltage drop to the side branch. So this sister branch, this sibling branch, this sister branch experience almost isopotential voltage, almost no drop to the side branch because of the sealed end boundary conditions here. So it's a very short cable, it's a very short cylinder, this side branch, 0.25 lambda long, and in this short cylinder is no voltage drop, but there is a lot of conductance drop. So if there is a synapse here, the conductance change in the side branch is not well seen. And it's symmetrical, so the voltage drop from, if these two dotted branches are identical, the drop from here to here, percentage-wise, is exactly the drop from here to here. But these are two symmetrical branches. Okay, so these are interesting properties because it says that in terms of conductance, the side branch is decoupled, is isolated, is not affected by the conductance change at the distal branch, unlike voltage.
So, after the symmetry here, if there
Oh,
was,
sorry.
so you have these two branches
Yes.
up there. If there will be a check, if you will switch the roles,
Yes.
you will have GIs at the other branch, and they're not the same branch.
Oh, they are not the same branch. Here they are the same.
But if they are not, it will not be symmetrical.
It will not be. No, if they are not, sorry, if they are not, in terms of voltage, it will not be symmetrical always. If they will be symmetrical.
Even if the
Yes. Yes. Any two ij that you take in a tree, any two I.
So here in this particular one, the same fraction of.
Yes, the same fraction. So the fraction from here to here, percentage-wise, is exactly the fraction from here to here, the drop, the percentage-wise drop. I also didn't look at it. It seems as if in my eye, I checked it analytically, numerically, it's the same. And it should be the same for any points in the tree. It's very hard to see by eye, but it is, I can assure you. Okay, I'm jumping about the explanation, but I want to come to the first surprise. So the first analytical surprise, or our surprise. So we took the case, so this is for a single input perturbation. We took the case where you have a nonlinear hotspot in the dendrite. You know the dendrites, especially of pyramidal cells, layer 2, 3, and layer 5, there's a lot of hotspots, a lot of active regions. NMDA spike, calcium spike, sodium spike. These dendrites are not passive in case of layer 5 and layer 2, 3 pyramidal cells at least. Also in terms of Purkinje cells. So let's speak about hotspot. This hotspot has an active current, let's say NMDA current. You activate synapses here, and you boost the hotspot. You use the hotspot. You get nonlinearity here. The question is, where will the synapse be most effective? Inhibitory synapses will be most effective in killing this hotspot, dampening the hotspot. Okay? The classical view is that it's always better to put the inhibitory synapse between your active current, this synaptic current, and the soma. This is the on-path condition that I mentioned before. But I want to show you the surprise, and now we understand it analytically, why actually if you want to dampen this hotspot, you better put your synapse either on top of the hotspot, this is always the best to really kill the hotspot before it grows. Before it becomes active, you shunt it, that's the best. But if you ask if I have the same distance backward or forward, so let's say some d from here to here or from here to here, I can prove mathematically that it's always better to put your inhibition distally to the hotspot rather than proximally. Just to show it graphically, so in this case, we only activated this inhibitory synapse, not this one. The hotspot is active, NMDA current is generated, the cell body is firing because the cell body has sodium generation spike, and the inhibitory synapse is active on the path, and that's what you get from the output. Of course, if there would be no inhibition, there would be more spikes here. So some of the current, of course, is shunted by this inhibitory synapse. But if you put now not this brown synapse, but the same synapse, inhibitory synapse distally, that's the output of the cell body. So you can see that the identical distal synapse is better in shunting the hotspot than the same proximal synapse. That's a surprise. And the answer lies there, because you can see that the boundary conditions which are sealed here and are leaky here gives this asymmetry of the shunt going, or the voltage attenuation from here to here backward. So the spread of shunt from here to here, the spread of shunt from here to here is less than the spread of shunt from here to here, just because of the difference in the boundary condition. And so eventually you get more shunt locally from this synapse at the hotspot. You get more shunt than from this synapse at the hotspot. So this synapse is more successful in shunting the hotspot because it is close to the boundary, to the sealed end boundary conditions.
How close should it be?
So we have a graph showing that this is always the case. So if you compare two synapses that lie in the same distance and you move them away, it's always the case.
Even if the seed at the end is very
If the seed end is very, very far, then of course they are the same, because the shunt is not felt at the hotspot and the hotspot is free to go to that. So of course, if it's very, very long both ways and the synapses are very, very distant, then the hotspot doesn't care about inhibition anyway, because it's already faded away.
I'm just saying, if the boundary, if the synapse is close.
No, if the boundary is far away, and this boundary is far away, so the boundary condition is not felt by the synapse, then again you are in the symmetrical case. So there are no difference anymore. But in a realistic sense, this is what we want to claim, that in a realistic sense, where you have a cable of 1 lambda long on average, dendrite, and if on a realistic cell you have a hotspot in some location, and we did a lot of computation, it's always better to put your inhibition behind. If your mission in life is not to kill the spike at the axon in the beginning, but also to kill the nonlinearity, to control the nonlinearity, to dampen the nonlinearity. So our claim is that much of these inhibitory synapses is designed to control local nonlinearities, and part of the fact that they are so distal is because of this. The distal synapses are effective. If I could really design, if I was a fantastic designer, and each time I have a hotspot locally, I will put exactly an inhibitory synapse on this hotspot, then it will be optimal. But this is not what happened in biology, as you know. This is beside the chandelier cell. Okay, but we want to solve the equation for the multiple conductances, because as I said before, there is never one inhibitory conductance. And I want to solve this, the cable equation for multiple conductances. We can do it iteratively, analytically, and we can also take cases like this, which are symmetrical and nice, and so we can compute, for example, for a starburst-like inhibition like this. We can show analytically that it's equivalent. This case, which is hard to solve analytically because you have many perturbation points for the cable equation, you can show mathematically that this case is equivalent to a single synapse sitting on a single original branch, and all other n synapses are sitting on what is called the equivalent cylinder of all the rest. So you close all the rest, represent all the rest as one cylinder with the particular dimensions coming from cable equation, and then you put the rest of the synapses as one big synapse here. And this case is exactly mathematically equivalent to this case. So I don't need now to solve for many locations. I can solve the cable equation for this particular structure with this particular boundary conditions, but for these two synapses, one big synapse representing n-1g and one synapse representing 1g. I can show mathematically that this is equivalent to this. And so, for example, if I want to see what will be the shunt level here, let's call it the soma. This is, let's say, the soma. What will be the shunt level of the soma as a result of these two synapses, or of these n synapses? I can do it analytically, and I can solve this. And here is the second surprise. This was really a surprise. I really did not believe Albert when he showed this, because it's absolutely against my intuition, completely against my intuition. and this is the following. So this is the shunt level at the synaptic location itself. When you have one synapse, two synapses, three synapses, four synapses, yes, you have one synapse on this structure, or you have six synapses in this structure, let's say. And you measure the conductance, the shunt level, with distance from the origin. This is the origin. And suddenly you see something that is really cannot happen with voltage, is that in the origin, let's say in this case, in this junction point here, you get more shunt, more conductance change at the shunt than at the synaptic site itself. You see, this will be at the synaptic site, and this will be at the middle. So that means that if I surround the region with synapses, inhibitory synapses, or excitatory synapses, with shunt, I'm looking only at the shunt now. If I shunt around the region, I suddenly may get inhibition stronger in the center than at the synapse itself. That's completely against the intuition of every biologist.
Come on, you have 6 synapses there. You can create a lot of charge through them.
Sorry?
Why shouldn't it be higher?
Because no, the question is, how does the shunt accumulate? So if you have only one synapse here, you see that the shunt, as expected, is always bigger here. If you would do it with voltage, if I will inject current here, if I will inject current, not conductance, if I will inject current I here, I will always get more voltage locally. Always. According to Ohm's law, I cannot get more voltage there when my current source is here. So it will always look like this. So voltage will be always larger at the input side. But conductance, no. So this is a surprise for me. I didn't think about it. Maybe it's not a surprise to somebody else, but I was really surprised that I can get a bigger shunt from encircling synapses at a location without a synapse. I always thought that near the synapse is the maximal effect of the synapse in terms of shunt.
All the synapses in the same place. It would be just 6 volts.
6 volts. Yeah, because conductance sum up. Yes, that's true. So locally, yes. Locally, as I said, if all the big synapses are local, the maximal shunt is local. But could I get a bigger shunt in another location just because you have the synapses encircling a region? I was not expecting to get a larger shunt at a region without synapses. I was not expecting it. Now I understand it, but I was not expecting it. You can also show the limit of it, so it's about 20 synapses can give you the maximal. Remember, we have about 20 inhibitory synapses surrounding the region in trees. Okay, so let me show you just graphically. Suppose you have 15 synapses. This is 5 synapses, another 5, another, I don't know, another 5. Okay? Or let's talk about 10 synapses, 10 here and 10 here. Let's say this is Martinotti synapses. And they occupy this region. So 5 here and 5 there. You can see, and this is the shunt level. So red is more shunt than blue. You can see suddenly the in-between region here becomes red. That means that the in-between region becomes shunted without synapses being there. I'm trying to say, because I won't have time to summarize, you can see already, I'm trying to say that if you want to control a region in terms of inhibition, you don't necessarily need to put each inhibitory synapse near excitatory synapse. Suppose I want to kill this region, inhibit this region, shunt this region, I just need to encircle this region. And then everything that is in between will be strongly affected, very strongly affected by this encircling, engulfing synapses. That's what I want to say. Okay, so let's go to the end almost. I want to show you, what do I want to show you? How much, 5 minutes or what? 10 minutes. Okay, so let's see if all this analytical game is relevant in some sense to some biology. So let's look at, again, I like to talk about Martinotti because we have a lot of data from Gilly, Silverberg, and Henry about Martinotti cells and the connection between Martinotti, which is an inhibitory cell, to layer 5 parameter cell. This is one example of reconstructed pair that I mentioned. This reconstructed pair in this particular case had 12 synapses, inhibitory synapses distributed like this. We have several like this. For example, this is another pair. This is 4 pairs. So you can see this is the domain of Martinotti. You can see domain of Martinotti. Martinotti cells typically converge on the apical tuft of layer 5 pyramidal cells, and also sometimes on the oblique branches here. So that's the typical domain. It's not a very narrow domain, but they never do on the basal tree, for example. So this is a large domain, but a domain. So we have a lot of information about that, and I think we did for the first time a very systematic study. This is a previous work with Albert, because Gilly succeeded to record from the cell body of the Martinotti cell the iPSP following bursts of spikes in the Martinotti cell. So the iPSP as the pyramidal cell looks like this. So there was a spike in the Martinotti, another spike, another spike, another spike, another spike. So this is the depressing iPSP, depressed iPSP. And so we try to, using the model, the detailed model, we try to estimate what is the conductance change, assuming that all contacts have the same conductance change, and that all of them are active. We can also assume some probability of being active, but inhibitory synapses are rather strong in their P, close to 1.8. So we can estimate the conductance change per contact that will give you, for this particular tree and this particular location, this iPSP. We can also, the Mishat-Sodix Henry Markov equation. We can also compute other parameters for the dynamics of this synapse. I'm not going into it. But since we have quite a handle on the strength of the Martinotti conductances, just because of this, I can then ask the question about what would be the expected effect of Martinotti over the dendritic tree. So, for example, if you have a single Martinotti, only a single Martinotti converging on the dendritic tree, which is not the case. There are about 5 Martinottis converging on a single dendritic tree. But if you would have only one, in this model, the shunt level will be like this. Now I'm using the real conductance change per contact, because now I have measured it, or at least estimated it. So you can see that the Martinotti cells tend to make this region red. This region becomes red. This surrounding region becomes red. Sometimes also there, depending on exactly, but this, and we already, when we saw that, we realized one thing immediately, that Martinotti cells controls the backpropagating action potential. And also controls the backfiring. So if there would be a calcium spike here, the redness here will decouple the calcium spike from the soma, and if there would be a spike at the soma, the Martinotti cell will decouple the backpropagating action potential just because of this shunt level. We also showed it experimentally later, and I don't have time to show it. We worked a lot with Matthew Larkum, and that's interesting that Martinotti, we know today that if you activate one pyramidal cell, that is called the Martinotti loop. If you activate one pyramidal cell, this one pyramidal cell recruits about 5 Martinotti cells, which all converge into the other Martinotti pyramidal cell. And we know that per loop, from 1 to 1, there are about 5. Total, they may have about 12 or maybe even 15 Martinotti converging into one cell. And we now understand much better, I think, both due to the theory but also due to the experiments, that the role of Martinotti really, also experimentally, is to block the calcium spike activating the cell body. So I'm going to jump on that, and I don't want to show it. Also in the model we can show it. And just to, because I don't have much time, just to summarize some aspects, then maybe I will say one word about spine. So we know that the pyramidal cell, we should not think about pyramidal cell as having one single ultimate output site. We may think about this like this, but between this ultimate output sodium spike, there are many nonlinearities in the dendrites. Some of these nonlinearities, like calcium spike, were already discovered or shown early on. I don't remember if it was on the 19th, Mike and Yael already showed these nonlinearities, but today we also know that there is a very strong NMDA local calcium spike. And so the feeling now that I have, that's an answer to question number 1, why do you have, or number 2 it was, why do we have this regional inhibition, is I think because we want to locally control nonlinearities. Maybe locally here by Martinotti and double bouquet, maybe locally here by the basket cell, maybe very locally here by the chandelier cell. But we certainly don't want to put inhibition only here, because if I would put inhibition only here, then all these nonlinearities will become free, will do what they want. I cannot control for local inhibition, let's say chandelier, this NMDA spike. But if I want to control this NMDA spike, for example, due to plasticity, I don't want this tree to be plastic. I don't want this tree to be too powerful in terms of conveying information, then I need to put locally. Not one by one, but locally, I need to cover this region. When I say cover, I mean to encircle it by enough inhibition to do this trick that I mentioned before, this enhanced centripetal or central shunting inhibition. Finally, I just want to comment on that. This is what I started, and that's because I'm working on this right now with Albert and another student on this issue of why they come together. So the classical notion is, the answer to this by Rodney Douglas, Kevin Martin, and other theoreticians is that basically this inhibition controls very, very specifically only this excitation, because it's so local. This is exactly not the case, as I showed you here, because the same inhibition that makes very local inhibition on this spine also makes inhibition on the base, so it's not anymore local. So this is certainly not the reason, only reason. Could be one possibility, but not the only reason, because the inhibition, as I said, covers a whole region. It doesn't only cover that. One thing I can say, I don't have time to show you, and you understand that, because the spine head is a very, very small compartment. The spine head is a very, very small compartment. This is about 0.2 micron in diameter. This is about half a micron in diameter or so. It's a very, very, very small compartment with an estimate input resistance locally of several gigaohms, maybe a gigaohm. It's a huge local input resistance just because of morphology. This means that a shunt, local shunt here, local conductance change here, will locally have a large shunt level. Just because you are now fighting, so to speak, with it's easy to fight with a region with a very high input resistance. You just need to change locally a little bit the conductance, it will shunt the synapse. So in this case, it's true. The local synapse will suffer a lot. The local synapse will locally suffer a lot from this local shunt. The local nonlinearities, the NMDA nonlinearity, the calcium flow, whatever the nonlinear processes are here, will very, very much be affected by the local inhibitory synapse. So that's true. Classically, the idea that if you want to control local synapse or local nonlinearity in this very, very, very isolated piece of dendrite, you should put an inhibition there. That's fine. But if you ask from the dendrite point of view, then you can show that if I compare the conductance, suppose my inhibition is here once, and then my inhibition is there, the same inhibition, once here and once there, and I measure the shunt level locally here. How much do I lose, so to speak, by putting the inhibition here, in terms of the local shunt level here, compared to the case where I place my inhibition there? We can prove that it's very, very, very little. It doesn't really matter from the point of view of the dendrite, whether the inhibition is here or here, unless you have a very, 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 very thin. By the way, these numbers today, there is always a competition between Karl Svoboda, who thinks about this, and recent measurements, who thinks about this. I think Karl Svoboda is right, and we are about here, or maybe 100 megaohm. So this is not such a big isolation. So the shunt here is felt here, and I don't have time to show this to you. I just want to summarize why the 6. Why cover the whole dendritic surface? We may discuss it. To control dendritic nonlinearities, I think that's the most important thing of having local inhibition distally and also dendritic plasticity. And I think that's why we call for a dendrocentric point of view rather than a somatic-centric point of view, at least because ולו בגלל we have dendritic nonlinearities locally. Why target specific dendritic domains? I think there is some kind of matching between regional nonlinearity as a global thing. Let's say the calcium spike is mostly at the apical branch of layer 5, then you need to put something locally there, at this particular subset of inhibitory neurons. I answer that. Why multiple? Because I want to make sure that I engulf a region. When I want to do something, I need not to have one inhibitory synapse. I need to engulf a whole region to control this region and make sure that if some of my synapses fail, I still succeed to control this region. Why so rich repertoire of firing? This is my question. Answer. I don't know. Really don't know. How does it affect the dynamics of the network? I think there is something probably extremely wise. You want the network to be, the output of the network, which is due to excitatory cell, to be reaching its output repertoire. You could decide to put a lot of effort on the 85 or 90% of cells with a lot of channels there to make the output cells very, very, very rich. But you could be a clever designer, that's a tentative answer, to put on the brakes, to make the brakes rich, to enrich the brakes rather than enrich the direct output. So you save a lot of synapse. So you save a lot of channels, ion channels, by putting them on a minute number of cells. These cells control the local dynamics, and the output is just expressing what the inhibitory cells are telling them. But that's just throwing hands, waving hands. I have to prove that the richness of the dynamics of the network is extremely, very much affected by the richness of the small inhibitory neuron. But that's my conjecture. Why 1585? Because you don't need one by one. 15% will cover well the 85%, and the spine issue is something I don't know as yet. So I think maybe that's enough. Maybe the thank you disappeared, but thank you. Maybe we need light.
A comment, and is it in single neurons? Is there a similar case with potassium and sodium? Actually, that the inhibitory potassium is much more varied and different subunits and
You mean the GABA A and GABA B?
no, potassium and sodium channels. Let's say the excitatory and inhibitory component of the single neuron. So I think, from an expert, that there is many more types of potassium than sodium.
It's one okay, I don't know if that's what you mean. That's what you mean, but it's true that in terms of richness of ion channels, the dendritic tree is the real interesting soup of ion channels. The initial segment is also rich in terms of different types of sodium ion channels. Mike can tell you more. But in terms of, if you ask me, in terms of investment of ion channel types, potassium, for example, there are several types. Calcium, for example, there are several types. There are today, I think we now recognize about 50 types of different ion channels in terms of voltage sensitivity and kinetic. Most of them are in the dendrites. So something in the dendrites, not only in terms of so part of the nonlinearities of the dendrites are coming from these channels. All these nonlinearities, NMDA nonlinearity, sodium nonlinearity. So most of the channels, the interesting channels are on the dendrites. Again, why I don't know. If that was your question, yes. Sodium is more limited to the all or none effect, not really almost, but mostly on the axon. The dendrites are more kind of more dynamics, more nonlinearities, rich, and they can shift from one location. So I think the dendrites are the interesting region for expressing nonlinearity in a gentle way. The axon is more all or none type of. Not exactly. I'm a little bit I'm putting the axon down. The axon also can be modulated and so on, but I think the dendrites can be modulated because of the richness of the ion channel there. By the way, your brain is mostly a dendrite, percentage-wise, yes? About 60% of the area of your cortex are dendrites, not axon. So maybe it's good to put some effort on the dendrites. I don't know.
I have just two suggestions. One is considering the spine. Let's say you have a memory you want to keep in the spine. So if you get the input, but you don't get the backpropagating action because you shunted the whole dendrite, so you want to save the memory, right? If you don't get the backpropagation, you will get LTD by acidity. Okay? So let's say you want to protect this memory. What should you do? You should shunt the inputs to that specific spine. That is a very precious memory. That's one wild suggestion.
But at some point, this memory should be expressed.
Yeah, yeah.
You want this memory to be expressed.
Not with shunting I mean, if you want it to be kept safe, you don't want it to be modulated. I mean, if you don't get the back propagation, that's one.
Yes, well, the back propagation, you enter into the back. You see, we have a lot of, we have a huge work, and I'm not going to talk about it, but we have a lot of work, we have a whole paper about the interaction between inhibition and excitatory plasticity. We can show what a single inhibitory synapse can do to the plasticity of excitatory synapses on the branch. We are using calcium-dependent plastic rule to model this plasticity by Shuval and Cooper. We're using this rule, which is a calcium-dependent plastic rule. This is a protected region. This is the LTP region. This is the LTD region. This is the LTP region. And we can show what will do particular inhibitory neurons on this plasticity. This is a whole paper I didn't have time to talk about. The interaction between synaptic inhibition and dendritic plasticity. And you can show that an inhibitory synapse can really dissect the tree into region of protected synapses, depressed synapses, LTP synapses, in the same small dendritic tree. But I didn't get time to talk about it. Without the backpropagation, it's a local, without the backpropagation.
But let's say within an active region, within a region you want to have plasticity, you want to save that.
To protect this region.
So you can do that.
So you can see that you can protect a region by the chandelier cell. All the synapses in this region, when the chandelier is active, all the synapses will be protected using this algorithm. But I didn't have time to talk about plasticity, so sorry about that. I thought that an hour and a quarter and a half is a lot, or an hour and a half is a lot, but it's not. It's always not enough.
So the second wild thing is actually related to what Mike asked you, is whether inhibitory cells can change the electrical property quite quickly from one pattern to another. And if you assume so, then let's say it's much more conserved to have just 15% cells that can change their channels, not what channels they have, but
you can block or you can modulate the channel. You can modulate the channel due to a hormone. And then you go to sleep and then you go to another activity. Yeah, you can do it very fast.
Much more economically efficient.
But why to do it only on the 10%? Because of the economical efficiency we spoke about.
Let's say that each channel you have to switch, cost you something.
Yeah. But before modulation, the fact that you have a 50 or 10% cells that are rich, and they influence the whole dynamic, and I have to prove. That's what we just now do in the Blue Brain project. We have the 10,000 cells glued. We have the types. And I'm asking, let's say that I'm closing basket cell, layer 2, 3 basket cells, how will it affect the dynamics of the network as a whole? And then maybe to have some measure about efficacy of this, so to speak, control versus another type of control. But we didn't do it. But my hunch is that the designer really took a small number of cells and put a lot of effort to make them rich in electrical properties, to make them rich in anatomical properties, to make them rich in connectivity patterns. A lot of work was placed into these inhibitory neurons in terms of some type of richness. But my theoretical question that I didn't show you is computationally, what is the advantage? And for this I need your help. Not the biophysical aspect, but the computational aspect. So these are just words. I have to show it mathematically.
There is a layer of complexity you didn't mention, in that there are inhibitory interneurons which selectively inhibit other inhibitory neurons.
That's true.
And these cells, especially the parvalbumin cells, which are fat-spiking cells, they are very specific spiking characteristics. Have you thought about, or have you modeled at all, what might happen in the inhibitory cells?
Not yet, because now our target cell was always excitatory,
right.
And we tried, because there we have a lot of anatomical and distribution data and so forth. We just, because of the pair recording, was mostly done inhibition excitation. Now, because of the Blue Brain, because we composed and put things together, and there are some inhibitory recordings, we can really start to build a better knowledge about the loop. Martinotti loop is part of this loop, one inhibitory loop. But I don't know, I don't even have an idea what will happen if I will inhibit you and you're on the other side and you will inhibit there. What will happen to the dynamics of the network? I have no idea. So there are these questions, I don't have an idea. I just wanted to highlight the importance of inhibition in terms of what I've learned through the process of becoming acquainted and even a friend of the inhibitor in Europe.
I think we're 10 minutes behind schedule, so
Thank you very much.
Continue offline.